Now we are going to discuss about gibberellin hormone. Contents of gibberellin hormone, history, physiological effects of gibberellin, and mechanism of action in gibberellin. The gibberellin was discovered by the same period of vaccine. In 1926, Japanese scientist Ichikurosawa identified that foolish shielding disease was caused by the fungus Gibberella fujikuri. The pedestal is thin color with thin pale color appearance, which is called Beckenham disease. In 1935, Yabuta was isolated the active substance that heat stable and gave the name Gibberella fujikuri. In 1938, Yabuta and Suzuki was isolated and identified gibberellin is a crystalline form and the gibberellin A, gibberellin B from their original preparation. In 1955, Brain et al. Was obtained by single gibberellin which is named as gibberellic acid. In 1960, Macmillan to be identical with gibberellin 1 earlier from Gibberella fujikuri. It is now known that there are 125 gibberellins which have been isolated under chemical properties. This is the structure of gibberellins. Next one, physiological effects of gibberellin. First one, seed germination. Second one, dormancy of foods or dormancy of buds. Third one, root growth. The gibberellin concentration is present on the plant. First, initially for longer root pod developed and laterally clusters of root pod is present, and it is a highly concentration of gibberellin is also present. Next one, elongation of internodes and blotting and flowering, induced parthenography or development of seedless fruits, and the next one, promote flowering in long leaf plants. Next one, biosynthesis of gibberellin. Gibberellins are usually synthesized from methyl erythritol phosphate or malic acid pyrophosphate pathway. Acetate is converted into acetyl-CoA. Acetyl-CoA is converted into HMG-CoA with the help of the enzyme acetyl-CoA transferase and HMG-CoA synthase. HMG-CoA is converted into malic acid and it is to NADPH2 is converted into to NADP and removal of two hydrogen molecule with the help of the enzyme HMG-CoA reductase. Malic acid is converted into malic acid 5-pyrophosphate or malic acid pyrophosphate, and it is 2-ADP is converted into 2-ADP molecule with the help of the enzyme malic acid kinase or phosphomalonate kinase. Malic acid pyrophosphate is converted into isopentenyl pyrophosphate and removal of carbon dioxide and water molecule with the help of the enzyme diphosphomalonate decarboxylase. Isopentenyl pyrophosphate is converted into dimethyl pyrophosphate with the help of the enzyme isopentenyl pyrophosphate isomerase. Isopentenyl pyrophosphate which having 5 carbon atoms is combined to dimethyl pyrophosphate which having 5 carbon atoms to form geranyl pyrophosphate which contains 10 carbon atoms. So it is called as monoterpenes. Geranyl pyrophosphate is combined to 1 isopentenyl pyrophosphate to form farnesyl pyrophosphate which having 15 carbon atoms. It is called as sesquiterpenes. The sesquiterpenes or farnesyl pyrophosphate is combined to 1 isopentenyl pyrophosphate to form geranyl pyrophosphate. It contains 20 carbon atoms. It is called as diterpenes. The geranyl pyrophosphate is inhibited ammo 1618 and phosphone D to form copolyl pyrophosphate. Entigerin is formed in the copolyl pyrophosphate and inhibited by phosphone D. The entigerin is converted into gibberellin 12 aldehyde and inhibited by paclobutrazole. The gibberellin 12 aldehyde is converted into gibberellin 12 and gibberellin 53. The gibberellin 12 and gibberellin 53 is combined to form all other gibberellins. Next one, mechanism of action in gibberellins. alum details for mobilization of endosperm food resource. The gibberellin combines with a receptor on the outer surface of plasma membrane of polyuron layer cells. The gibberellin receptor complex interacts with a heterotrimeric G protein and initiates two separate signaling transaction pathways. First one, calcium dependent signal transaction pathway, and another one, calcium dependent signal transaction pathway. The calcium independent signal transaction pathway, which involves cyclic GMP as a signaling intermediates leading to the expression of alpha amylase gene. The calcium dependent signal transaction pathway, which involves calcium binding protein, convolutin, and protein in kinase signaling intermediates leading to the stimulation of alpha amylase and other hydrolytic enzymes from cells of polyuron layer into the cell endosperm for starch degradation. The gibberellin signaling intermediate is activated. The activated gibberellin intermediate goes into the nucleus and binds with the delta receptor or receptors proteins and also spy are otherwise called as spindle mutants. The delta repressor and spy are degraded or inactivated. Due to inactivation or degradation of these repressors, gibberellin, MIB and other genes are switched on. The gibberellin MIB mRNA goes into the cytosol for translation and the gibberellin MIB proteins is synthesized or transcription factors are synthesized. Gibberellin MIB proteins enter into the nucleus and binds with the promoter genes for alpha amylase and other hydrolytic enzymes are formed. The alpha amylase and other genes that encode other hydrolytic enzymes are transcribed. Alpha amylase mRNA moves out from nucleus to rough endoplasmic reticulum in cytoplasm for translation process and alpha amylase protein is synthesized. Alpha amylase proteins are secreted via Golgi bodies. Thank you.